My name is Elliot Ragano indeed, and I am the technical lead of IntAct Complex Portal and Reactome. And so with me is Suzy Huger, who made the biggest part of the presentation today, and will present the second half. And she was a part of our team, but she's now working for IME, a French company. So today we're going to try to explore the molecular interactions in depth and across species thanks to IntAct. So if you want to access the website, this is the URL to access it. So ebi.ac.uk/intact, and you can follow on what I'm talking about while I'm speaking. So in terms of definition, within IntAct, one node will represent one molecule, while one edge is one interaction evidence. That is to say that it is one interaction report observed by one technology in one publication. And the technology can either be, is both the interaction detection method, but also the interactor detection method. So that is to say, if you have one publication which has two different experiments showcasing molecule A and B interacting and molecule B and C interacting, that will be two distinct interaction evidence. But you could also have protein A and B being shown as interacting together in two different publications, and these will also count as two different interaction evidence. So then this is what a typical network within IntAct looks like. You have nodes, you've got edges which are binding those nodes together and representing those evidence of interaction. So this data is manually curated by our team of curators in IntAct. They take literature examples, so they look at all the literature and extract publications of interest, which contain data related to molecular interaction. Out of that, they extract the experiments and specifically which interactions involving which interactor, and down to the level of features. For instance, among features, we have the mutation. So if one of the participants of the interaction was mutated, then that information is recorded within the database. And then we also assess the impact of that mutation on the interaction. So we have really an in-depth data model, and that is all then being validated by a reviewer afterwards. And upon approval, it is then released onto our database. But we're not the only one who are doing this. We're actually part of a consortium called IMEX, which aims to standardize and has created consensus on what is the data models and data formats that we are using to describing interactions. And this IMEX is really about sharing resources, and all the IMEX curated data is then imported within IntAct to be delivered to you as efficiently as possible. And also Psychic is a querying software which is emerging from this IMEX consortium. So in terms of statistics, within IntAct at the moment, we have 23,000 different publications that has been manually curated, covering more than 145,000 interactors. And there are, of course, quite a lot of redundancy between them. So we have a lot more interactions than interactors. So in terms of pure interaction, we have 871,000, but when you expand that to binary interactions, which I will explain just a bit later, you reach 1,700,000 different interactions. In terms of coverage, and by coverage, we mean how many nodes from a proteome are being touched by at least one edge within IntAct, also which is interacting with at least one interacting partner. So in Homo sapiens, close to 90% of proteins from the reference proteome have been covered. And same for Saccharomyces cerevisiae, as you can see here. And Drosophila melanogaster, we have really high coverage for those three species, but we are not limited to those species. We actually cover most species. It's just happened that there is more research effort produced in those organisms. So that's why we observe them more as well. But we have a pretty solid coverage, and we are aiming at extending that as much as possible. Okay, so now let's see how you can understand the network that we built. So this is a typical result that you get from, in this case, querying for exokinase 1 within IntAct. You can see that you have many different nodes of different colors and different shapes and different edges. So first, let's go through the color nodes. Not colors, sorry. So most of them are light blue. This is the Homo sapiens, and yeah, they are representing the vast majority of nodes, as you saw just before, within IntAct, and they're this color. Then you've got Mus musculus, which is slightly darker, and then other mammals, which are in brighter blue, as such. Then this is telling you information about the species, but then you also have information about the data type. So are we talking about a protein interacting with a protein? Are we talking about a gene interacting with a protein, or a protein with some nucleic acid? So yeah, all of that is encoded by the different shapes of the nodes, as you can see here. Finally, this is what we call the summary mode, because all the different evidence of interaction between two participants, let's say between CFTR and ATP2A2, are gathered in a single edge, which is summarizing all the knowledge that we have about those two proteins interacting together. And for that, we have the color is encoded by the MI score, which is giving a clue on the confidence that you can put into these two proteins interacting together. And the thickness of the edge is summarizing how many evidences we have that those two proteins are interacting together. So they're kind of related to each other, but these are the two pending aspects on the summary mode. Now, if you go to the evidence mode, we are suddenly expanding all those summary edges into many different evidence edges. So then suddenly every single edge represents a different interaction evidence between two proteins or two molecules. For instance, here we had previously just one, and now it was actually summarizing 6 different edges. And you can see as well that they have different colors. The colors are dependent on the type of the interaction. So it goes from a really weak colocalization. Basically, two proteins were found close to each other under a microscope, and we say they're probably interacting together. That is a relatively weak level of evidence. But we have much more stronger type of interaction evidence, like the direct interactions, which are giving more trust to those two proteins really interacting together. And so this gives you a kind of gradient in terms of intensity of edges here. And then finally, we have dashed lines here whenever the interaction evidence is coming from a spoke expanded model, which I will show right after. Finally, the last mode that is available is the mutation mode, in which each evidence of interaction is still represented by one edge, but it is highlighted in this pink color whenever it is one of the participants of the interaction has been mutated. And then that means that you will get some more information by looking in depth at it to whether this mutation had an impact on the interaction or not. And you can see both the edges and the mutated protein in the interaction partners are being highlighted as such. So in this interaction evidence, for instance, this is piezo1, which was mutated. No, the MI score is, as I said, a confidence score telling you how confident you can be that the two proteins are interacting together. And to get to that score, what we do is that we take all the publications concerning those two proteins interacting together, then all the different detection methods that were used to find those two proteins interacting together. And finally, what were the interaction types? And for detection methods and interaction types, we associate to them different weights depending on the level of quality that those methods or interaction types have. So we give more weight to strong evidences and lesser weight to weaker evidences. Then we normalize all of that so that we achieve a score between 0 and 1. And this is the scale that you get out of it. This is the global distribution of scoring across IntAct. And as you can see, the vast majority is hold over there. But what we recommend to have a high level of confidence within the interaction data that you observe is to put the threshold at 0.40, and then you have thrown away all the anecdotic pieces of evidence to really focus on what has been strongly observed multiple times. Now, the NRE expansion model. So sometimes some experiments that we curate are not talking about protein A interacting with protein B, but are instead focusing on one protein, which we're going to call the bait, and we throw at it a bunch of praise proteins, so other type of proteins, and we basically see which one sticks to it. And this tells us about a complex formation between all those different proteins, but we don't know necessarily which one is interacting with which exactly. We just know that they're all together interacting together. So when we have that, we have two ways to modelize that within a graphical, like graph theory modelization. The first way is the matrix expansion. Basically, you consider that every single participant is interacting with all the other participants. The other way is a Spock expansion, where you consider that only the bait is interacting with the praise, and you don't consider interaction between the praise. What you might observe from this is that there is much, much less edges produced by the Spock expansion than by the matrix expansion, because as n grows, this is pretty linear. If you add a protein to the complex, you have just one more interaction, but here you start to get exponential growth as n progresses. So let's try to compare and evaluate those two modelizations. Let's say that we have, in reality, this complex being formed. So the bait was interacting with those four proteins, and these other praise were interacting as such. If we try to measure how much positive and false negative we have, this is what we get. As we can see in the matrix expansion, of course, because all the possible... Sorry, I don't know what happened. Okay. All the possible edges that could exist are present, then there is no false negatives because there just cannot be. However, the numbers of false positive is really high, 32, and that will be even more of a problem as n progresses. On the other hand, Spock expansion, of course, because we lacked all those different interaction here, it actually have quite a lot of false negatives, but the numbers of false positive is drastically reduced. And in general, the numbers of errors of those modelization is much lower for Spock expansion than matrix expansion. And as n progresses, in general, matrix expansion is becoming worse and worse, while Spock expansion holds pretty tight to the reality. So that's why in IntAct, we mostly, or if not exclusively, use a Spock expansion model, but because it can indeed contain false positives and false negatives, we are representing them with dashed lines as such. And you can easily filter them across all the different platforms in which IntAct data is hosted, so that you only get really proved binary interactions if you wish so. So in terms of numbers across the 1,700,000 total binary interaction, 47% were true binary from single experiments, but 53% came from expanded interactions, and in total 61,000 NR interactions, which means that we have an average N of close to 15, and in terms of range, it goes from 3 to a gigantic 2,000 interactors interaction. So this one is really gigantic. So now that we have all those networks, you can then filter it to really focus on what matters to you. The first filters is about species. So you can filter for each species of nodes. So if you're only focused on human, you can focus to human, and on rat, or you can also get interested in infectious patterns, so include both the disease and the disease species and the infectious body, or infected organism, sorry. So that's possible. Another thing that is possible as well is to filter out cross-species interactions. So you can see here, for instance, in this specific example, that we had 225 different interactions involving at least a human, but only 180,000 81, sorry, my numbers are messed up, which are really between human and human interacting. The rest of them are human with another species, probably one of the ones below. So you can choose to focus only on human-human interaction by clicking on this, or you can open up to cross-species interactions by leaving that unselected. Okay, now then you have the interactor type, so you can focus on different molecule types, protein, mRNA, gene, stable complexes, nucleic acids, small molecules, and some more as well. You have also different interaction type, which you can filter by. As I said, you can only include if you want, for instance, the strong interactions, or you can, yeah, choose to only look at colocalization if you want to. In terms of detection methods, so there's quite a big diversity actually of detection methods, but all of them will be referenced here, and you can choose if you want to exclude or include any of them over there. You have also the interaction host organism. So actually, most experiments are performed either in vitro or in a specific cell line of a species, and sometimes like the fact that it is expressed in a human cell will have some impact on the interaction, or the fact that it is expressed in such type of cell can have an impact. So this is really helping you to give more clue on the context in which this evidence have been discovered, and filter out for things which are more relevant in your context. For instance, if you're looking at cancer studies, it might be more relevant to study the carcinoma cell lines and how proteins behave in an actual cancerous cell. Then you've got the mutation, expansion, and positive filters. These are pretty straightforward. So for the mutation, if you activate it, you only remove every non-mutated interactions or non-mutated proteins. Then you can also filter the non-expanded interactions so that you only have the true binaries that I was explaining earlier. And finally, you have the positive and or negative interactions only. This is actually quite complicated, but so in the vast majority of IntAct, most interactions are about saying, okay, molecule A is interacting with molecule B. But sometimes we have proof that A is never or not interacting with protein B in a specific context. And to annotate such type of data, we have this little cross on the edges. However, since it's quite confusing to describe what does not exist, by default, we are filtering them out. But if you want, you can always choose to include them if that's of any interest to you. Finally, you have the My score, which allow you to filter, well, based on the scoring system and the confidence system, both on the minimum and on the maximum. Then you can export the visualization that I showed you earlier, so the visualization itself using the network export, either as an image or as GraphML for networks import. Or you can also download the table representation of the network, either in the MyTab format, which is the most human-friendly. And as you progress within the versions, it gets more and more complicated in terms of how many columns you have. But then you have the choice to choose a really complete one or rather simple one with a 2.5. You've also got the AMI-XML, which is more probably best suited for computer analysis. So this is really the most complete format that we have. It contains absolutely all the data that we have, but it's quite difficult to interpret as a human. So yeah, that's a good format still. And then we have a slightly simpler format, which is the MyJSON, which is like, yeah, a simplified MyXML format, but which is also available if you need to have a quick overlook of the different interactions that we have within IntAct. Now, when you look at the networks, you have first the networks, but just below that, you actually have the different tables. You have a first table, which is the interactors, which is basically each node will have a different row there, and we will tell you all that we know about it. So its accession, its name, preferred ID, the protein type, the species of the molecule, a sort of description, different IDs in different databases, and how much interactions we know about it. One thing you might notice is that some, or if not all, of the terms here are represented with dashed line, and that is because they're actually controlled vocabulary, sorry. So you can click on any of them to get to know the definition of it and what we really mean by that term. And this is all maintained within an ontology system so that you always have a clear idea of what we are talking about and that things are standardized across the different IMEX partners. In the right here, you have the column toggle, which allow you to show less or more data according to your needs. Then comes the interaction table where most of the fun really happen. So this one is containing quite a lot more data about who is interacting with who, basically. And so everything that there is to know about the two participants, and as well like which role each of the participants were having, both experimentally and biologically, if they have any aliases, if they have features, parameters, annotation specific to that interaction, anything like this. So yeah, it's quite a complete table and a complex one, but you can control whatever you want to really visualize using this column toggle once again. Now, if we focus on the first one, you can see this is one interaction evidence or binary interaction evidence. It might actually come from an array interaction because we are expanding them through the Spock expanded. In this case, though, as you can see here, it's not expanded. So it's actually a true binary interaction. And you can check all the detail that we have about this specific interaction evidence by clicking on this button over there. And by doing so, you reach the single interaction view. So it has a little identifier and this really nice viewer, which allows you to see which were the different interacting partners and what were observed onto them. So for instance, by default, we are loading UniProt features on them. And you can see here within their sequence what is happening within the different domains of the proteins. You can also load MI features. That's where we are loading, for instance, mutation. You can see here that exokinase 1 was being mutated on 3 different places, and actually a 4th one over there. So there's 4 different mutations that has five, sorry, 5 different mutation that has been studied on this specific interaction, and I will see later what is the impact of that. So here in the details, you got a kind of summary of everything about this again. So the accession, the type, the detection method, the host organism, whether it's positive or negative, some cross-references to the different other databases and annotations whenever they were relevant. Also on the right, you've got this primary source of that evidence, which is a publication. So the title, the journal, the authors, the PubMed reference, and other cross-references. Then you have the participants table, which describe again what were the 2 different or many different participants in that interaction. What were their roles? So some were bait, some others were prey, etc., etc. So you have information there. And then finally, you've got the features, which I was describing a bit earlier and were represented visually in the diagram. But here you really get to see, okay, this protein is actually mutated on this localization, and that had was disrupting the strength of the interaction, while those mutations, on the other hand, were having no effect on the mutation. Sometimes, it's really rare, but sometimes they do create additional interactions, like de novo interactions because of the mutations. Also, here we can see that this post-translational modification is required for the interaction to happen. So this is like really valuable information that you get there. Okay, now how do you build all those networks and get access to all those details? Well, when you reach IntAct, the first thing you get is to this homepage and this quick search. Within this quick search, you can search for a gene of interest, a protein, or many other things. So gene names, taxon IDs, complex ACs, UniProt ACs, publication IDs, go terms, etc., etc. So when you perform a search as such, you will find a kind of resolution table because actually exokinase 1, you have one in human, but you also have one in musculus. You have also different isomers. For instance, this is an mRNA. This is like the main canonical one. So yeah, you have many different variation around hexokinase, and there you can either search for whole types of hexokinase or specify which specific hexokinase you're really interested into by clicking on the row constant. So for instance, this is a difference between loading all of the hexokinases and loading just one specific. You can see instead of having all those many blobs, you have just one corresponding to this human protein. The second type of access ways to build networks is using the batch search. In batch search, you provide a list of molecule identifiers or name. So let's say here 3, and then we will try to find within IntAct database which interactors you were really talking about. Then it's up to you to say, okay, what I really meant was this one and this one and this one and not the other ones. And then we will build a network with those different proteins or molecules. Then you have an additional option, which is whether you want to only focus on interaction within that set that you provided, or if you also want to include first neighbors, which are common or not between the set that you provided. So you can choose to include or exclude first neighbors as such. The last method, which is the more complicated but also the most powerful, is the advanced search, as it allows you to use a complex query language to do really complicated queries. For instance, you have, let's say you are interested in human SARS-CoV-2 interactions involving a mutation or of high confidence in general. So you could do it as such. You could say species Homo sapiens, and using the UI below, you can also actually select like it tells you what are the different fields available. And yeah, both fields are synchronized with one another. So you can say, yeah, species is Homo sapiens and SARS-CoV-2 because you have two participants. And then you have either mutation or high my score as such. And this will give you a results containing all the data that we have available corresponding at that exact query. Finally, we have two little ways to build, like we have pre-processes to build like complex data sets. The first ones are through curated data sets with Alzheimer, cancer. Whenever a curator comes across something that is known to be associated to Alzheimer or cancer or is somehow relevant for that specific disease, they will annotate it as such and it will be added to that data set. We have also computational data sets like virus, apoptosis, whenever one of the participants of an interaction is annotated as relevant for that specific topic, then it gets added automatically. We have also 16 species interactomes, and we have a mutation dataset covering all the different mutation across all species and what was the effect of the mutation on the interaction. And so, yeah, the last one is interactomes that I was talking a bit earlier. And within that, you can either click on the icon of the species to load the network in the website, or download the different formats available for them below. If you do click on the icon, though, because they are the biggest interactomes, so the biggest amount of data that we have in the data set, most of the time, if not always, you will have too many results to be displayed within the website. Actually, we have a limit of 1,500 different interactions that can be visualized in the website. But so in order to fix this, you can do two things. Either you can use the filters to reduce down the data to something more easy to handle, or make your search more specific. Or finally, you can load your data in Cytoscape. And that has two advantages. One, it can handle all of the data of IntAct, so there's no restriction in terms of size, but also it has a really large ecosystem of application that can allow you to do much more deeper analysis on the graph aspect of the networks. And that's where I will leave the way to Susie to present the IntAct app on Cytoscape.
Sorry, nothing to share. Okay. Good afternoon, everybody. I'm Susie. I used to work for IntAct and the Complex portal at the EBI. Now I'm a software engineer for the company in France. And so while I was working at the EBI, I worked for the IntAct Cytoscape app and also IntAct backend. So I'm going to talk about the Cytoscape app. So a bit of an introduction of the Cytoscape app. So as Elliot explained, we developed it to be able to overcome the limitations of the website. And also Cytoscape is quite handy when you want to personalize, like you can change the colors of the elements of the visualization, and also you can change the layouts. So for a bit of information, Cytoscape is developed in Java 17, and so the Cytoscape IntAct app is also developed in Java 17. It's using Java Swing for everything that is graphical. And so right now we are under the version of IntAct app 1.1.0. And so, as Elliot said, you can visualize, at least for IntAct, you can visualize an unlimited amount of data. So for example, here you can see on the right of the slide, this is the whole IntAct database. So there's absolutely no limitation. So let's start. So here you can see this window is the Cytoscape app. And so here on the left, you have a few panels related to the applications. And here you have the search bar. On the left of the search bar, you have a little button that you can click, and which will show you all the search bars from all the applications you have on Cytoscape. So for us, what's interesting is IntAct. So with IntAct apps, you have 3 types of search. You have the advanced search, the exact query, and the fuzzy search. We're going to start with the exact query. So basically, to make an exact query search, you need the precise identifier of the elements you want to visualize. So for example, here I wanted to visualize 2 proteins. So I've input the UniProt identifiers of those proteins, and it will display those proteins and their interaction as a network within the visualization window. Then we have the fuzzy search, which is a bit of a mix between advanced search and exact queries. So what it does, when you type in a query, it will take the terms of this query, and it will look for them within the IntAct database. And it will retrieve all the nodes that are related to this term. So when you click on search, it will display this window, which is showing all the nodes related to this term. So here I typed in NDCHE, so it will show all of them. And so you can select here whatever nodes you want to display. So for me, I chose to display Homo sapiens and the first neighbors. And so when you click on Build Network, it will show you all the interactions for the selected nodes. And then the last adding we did for the recently updated app is the IntAct Advanced Search. So the aim of it is to basically be like the search bar inside of IntAct. And so when you click on the search bar, this little pop-up will open. So here you can type in whatever you want to look for, or you can use a query builder. And when you click on that, a new window will open, which is containing a query builder, which is similar to the one on the IntAct website. So you can add your rules, your rule sets, and so on the left you have the element you want to filter. On the second row column, You will have all the parameters you can filter. Depending on that, you will have either operators, booleans, or ranges. And then you can type in whatever you want to filter. And then you can click on the Build Query button, which will launch the query and show the networks related to that. And so you can also type in, for example, here, NDCAT, and it will build the query. So here I hide here the search bars. So this is what you get when you make a visualization. So on the bottom, you will have tables related to this network. So you have a node table, an edge table, and a network table. And it's working like on the website. So on the node table, you will have all the information related to this, to all the nodes of the network. Same for the edges and the network. Then on the right side, you have everything related to the view and the filters and the different legends related to the network. So we will come back to the view part later. You also have actions here, so you can reset the filters, apply the filters on different networks. And also the extract for analysis button, basically it duplicates the network, so you can keep the one, this one, for example, this way and work on another network if you want to add some stuff, move some stuff. And then here you have 3 panels, so you have the legend, the nodes, and the edges. So the same way as we have on the website, you have the colors for the species. You have also the different shapes. Here it's not a good example because there's only one shape, but you have different shapes for the different types of nodes. And also you have the legend for the edges, their colors, and also the number of evidences. Then here, still on NDCAT, when we go to the nodes panel, you have all the filters. So here you can filter by type, species, and also when you select one node, so here this one is selected in yellow, you will have all the information about this node right here. Everything here is clickable. It will lead you, for example, for protein, to the OLS website for information about this term. Here for UniProt, it will lead you to UniProt, etc., etc. And so the same way for the edges, you will have all the filters on the top, and when you select an edge, you will have all the information about this edge. So here, since we are in summary view, you can click here to expand the summarized edge and see everything that has been collapsed into this one edge. And also you have informations about the interactors linked to this edge. Here we have zoomed in a little. So here right now we are in summary view, but the same way as on the website, you can have the evidence view, which will expand the summary edges. Also, we have the spoke expansion that we can filter here on the edge filters. And also we have the mutation view with all the elements affected by mutation highlighted in pink. So we'll zoom back a little. We are back in the summary view. And here is the orthology view. It's not really a view, and it's not really a filter either, but basically for the orthology, so a bit of history about it. Firstly, we implemented it on the complex portal, and the complex portal and IntAct databases are communicating within each other. And so for the orthology, what we did is that we imported data from the Panther database. So how it works in Panther, they are making those groups of orthologs and which are containing proteins. And so the proteins, they have their UniProt identifier. And so each ortholo group have a Panther identifier. And what we did on our side, we fetched the data and we compared our UniProt identifier, because the proteins we have are linked to UniProt. We compared all of our proteins to the proteins in Panther, and for each match, we retrieved the ortholo group identifier. We added it in our database as a cross-reference. And so how does it work in Cytoscape? For all the proteins we have in a network, we are using the Cytoscape. There's a built-in feature, which is the grouping, and you can group by something that is in the table. So we chose to group all the proteins by the Panther cross-reference. Also, when I did the implementation for the orthology, I also thought about the future. And for now, we only have the Panther data, but maybe in the future, there might be other databases that we might use. And so maybe one day we will be able to select which database we want to use for the orthology inside of Cytoscape. And so here, when we zoom a little, this is an ortholo group, so they are all in with this little green bubble here. Here I selected this one, so it appears yellow. And so when you click on it, in the nodes tab of the filters, you will have all the selected nodes info. So here the ortholo group is considered a bit of like a node, and so it will show you all the interactors that are contained in this group with all its information the same way it shows for the usual nodes. And also, yeah, this is a little reminder. So for the Panther database, it's only for proteins. And also, as I said before, the data has been imported on complex portals, so it's visible on the complex navigator. I have some slides for it if we have time, but I don't know for now if we will have time. If you are interested, I can show them. And so the last thing I wanted to present, it's a bit hidden, but it's quite useful, is the Cytoscape command line. So when you have Cytoscape on the bottom left corner, you will have this command line little button, which will open a window where you can basically type in what you want to do in Cytoscape. And so here, I tried to highlight in pink what I typed in. So here, here are the comments of all that are available on IntAct. And so for example, here, if I want to do an advanced query, I will just have to type IntAct advanced query equals, and in between quotes, what you want to look for. And so it will generate the network. And for example, if you want to add the orthology on it, you can just type in IntAct orthology, and it will group the orthologs together. And then you can also add different views. So you can also, for example, go into evidence view. I think that's all for me now. If you ever need any user guides, you can go to the IntAct website. There's a user guide section. Also, if you want to download the data or access to our API, you can directly go to the technical corner section of IntAct, which is linked here. If you ever need help, or if you want to give some feedback, you can send us an email to this email address. Also, we are on social media, so on Blue Sky and LinkedIn, and also Twitter. So basically we try to post regularly. So every month there's the dataset of the month. The curators are selecting one dataset they want to highlight. Also, we try to post when there are some events and we can find some data related to that. We try to do that. And also if there are big publications that are curated, the curators are usually posting about them in social media. So if you want news about IntAct in general, you can go to our social media. I would like to thank all the IntAct contributors and all the IntAct funders. And I think it's time for questions.
Oh, thank you, Susie. Sorry, my camera took a moment to switch on. So thank you so much, Elliot and Susie, for the talk. I forgot to mention at the beginning of the webinar, we are also joined by Kalpana, who is a senior scientific database curator at the molecular network team at EMBL-EBI. So she also helped with some of the questions that pop up during the talk. So we have actually a few questions that were asked during the webinar. All of them, I think, have been already answered. But since we still have 10 minutes, maybe to give us some time if someone wants to ask some questions now, there was one question that was particularly interested and Kalpana helped with the answer. But I think could be interested to bring it live because one participant was asking about, let's say, differences between using string or using IntAct and
Yeah.
when should I use one or the other. So maybe for all the other attendees, if we could also take this question live. Thank you.
Okay, I can start. String is a good app in the sense like when you want to explore interactions for the non-model organisms, and it gives a predicted information based on the conserved domains, interaction evidences that is collected from, primary databases like IntAct, and also from text mining evidences, post-translation modification, and other things. Okay, so then, so this is more appreciative because manual curation is very demanding task, and intact curation, which is backed by experimental evidences, it takes a lot of time. So we haven't got enough resources to cover the entire interactome, not only human, but also the other organisms. So that is where your applications like string can really help us to explore the uncovered interactions in across species. But whenever I have evidence, strong experimental evidences, I would definitely go for intact app. And also string networks give the beautiful 3D images, which I really adore them. So it's also that IntAct has its own Cytoscape app, and we have a different style of visualization. But I have my choices when it's based on the model organism. I mean, which organism that I want to explore. And so each has its own advantage and disadvantage. Yeah.
Thank you. I don't know if Susie or
Okay.
Elliot wants to add something on this, or we can go on to the next question. Good. Okay. Thank you. We have a question that was just asked and connects actually IntAct with other resources that are hosted at EBI. So I will read out the question for you. So since IntAct focuses exclusively on experimental validating molecular interactions and does not contain GWAS, EQTL, or SQTL data, how can we effectively connect IntAct with the Open Targets platform to achieve unified visualization and interpretation? Is it possible using Cytoscape?
Well, our data is already shared with Open Targets, and Open Targets has intact base evidences. I think it's in one of the tabs that you get. So you'll be able to fetch information. I mean, I don't think you have a direct option to get intact evidences from Open Targets and load into Cytoscape, but you'll be able to download the data from Open Targets and then relate it to Cytoscape. So basically, I'm saying that, okay, this is different layers of information, intact GWAS, EQ12 and SQ12. So there's we do not offer one pipeline wherein you can just directly bring in everything to Cytoscape. Right at the moment, it's difficult. It's very demanding. While running the resources, we understand it's demanding, so much demand on the developer side. And so as of now, you can get all the relationship via Open Targets and then get it downloaded from there and import into Cytoscape. That's the only choice I see right now. Yep.
Thank you so much, Kalpana. So if nothing to add on this question, we can move to the next one. So next one is also on Cytoscape. So when using the IntAct Cytoscape app, what is the best strategy or filter to quickly compare the interaction network of a protein, for instance, histone H3, versus a specific point mutated version of the protein? And the attendee is citing an example for histone H3 of a mutant.
I can take care of answering that, I think. So the first step would be to find the protein. So I'm not sure I will be able to find it. Ooh, H3 is quite big. Let's try to find the histone on Uniprot. H3 histone. Yes. Okay. Oh yeah, but that's on yeast. Let's say human, yeast 3. Let's assume this is the one you were talking about. So let's go within Cytoscape and query for it. Do we have... No, we don't have any information about this specific histone.
Would
Let's
you
just...
like to share your screen? I don't know if
Oh,
it can be
sorry, I thought I
useful.
was sharing my screen already.
Yes, no. I thought you were finding something and then you would link, but then when you mentioned Cytoscape, I said maybe it's better.
Okay, so let's try to find histone in general. So fuzzy search, histone. Okay, I don't know which histone specifically you were referring to.
So in terms of the histone was H3 and the mutation was H3K27M.
We have quite a lot of H3.
But I guess if it's something applicable, you can just kind of use another mutation. Let's say just
Yes.
to demo.
Let's try to load all the histones, I guess, from human. So we're creating IntAct and trying to build a network. Takes a bit of time. Well, and now everything is stuck. Okay. Okay, let's cancel this query. I'm gonna showcase. So let's say you manage to build a network. I don't know why this is taking so much time right now, but for time interest, let's keep it short. So the good thing here, so let's say this was your protein of interest. Because we are in summary mode here, you can see actually in this protein all the different mutations that we have inside of it. And so if you manage to find within it the mutation of your interest, let's say H3K27M. So it wouldn't be referenced as K27M, but using this code instead. And then you can see wherever that is happening. And as you can see here, for instance, this specific mutation have been observed on different evidence. So you can then select them here and it will, well, highlight them in this node table and edge table, and you can see all the information we have about them. And actually, if you scroll lower, if you go in the edge table, you then get all the information about the context in which this mutation was happening. So really, the solution would be to select the node of interest. So your hexokinase 3 in your case, open up the mutation tree here until you find the one of your interest and see all the different evidence of observation of this specific mutation. The other way, of course, would be to look within our mutation data set, which is on tabular format, and just look for the one of your interest and see what are the different things here. But within Cytoscape, this would be the way.
Thank you. There is another question that just came in, and it's quite detailed. But if I understood correctly, the meaning of the question in the context of the webinar would be that there are certain proteins, like the epidermal growth factor, that when are modified, like glycosylation, for instance, then they change the way they interact. So how is this addressed in the moment where I observe a network and I have a protein interacting with another one, when there is the modification, it changes.
So Kalpana, you can maybe answer this one.
Definitely. So as I understand, proteins undergoing post-translation modifications affects the interaction outcome definitely. And IntAct has been trying to capture these whenever we have these evidences. So as of now, we do capture them as the post-translation modification feature and add a definite rule saying that how it affects the interaction outcome, whether it's a prerequisite for the interaction to happen or it is an observed outcome of an interaction. So then the dynamics that it affects the downstream of the pathway. So that is something that we have to integrate to Reactome. So it's like, as of now, I would say IntAct has this feature tab where it brings out all the features that are affecting the interaction outcome, which includes the mutations and the post-translation modifications. And you can try to enrich your analysis using this feature tab. Maybe you can share the link quickly. Give me a second. Okay, so in documentation, where do we have the feature tab? Downloads. FTP site. We should, I think, where do we have the feature tab? Okay. I think you can use our FTP site here to everybody. Yeah. And I think you'll be able to access our feature tab through our download site. And then, so this contains the entire, it was earlier, we used to document only the mutations. Now you have not only the mutations, but also the other features, including the post-translation modifications with their impact they have on their respective interacting partner. So then you can enrich your network with these tab file formats on top of your network, and you can add them, and then you'll have the outcomes in your downstream signaling analysis. But directly, yeah. I think that's the best way going forward. Yeah.
Thank you so much. It's technically time, and we have only one question left. So if we can stay a couple minutes more, maybe we can take it. So it's quite a broad question. So someone is saying, basically molecular interaction can be quite messy. It's a very big universe. There are a lot of elements, etc. So how can we actually treat them and interpret them and study them in a way that is biologically meaningful and computationally tractable? So I guess it's like advice or tips or standard practice for someone that is in the field that you may want to give.
Thank you. Yeah, I think I'll take this. So as a member of, I've been curating for IntAct since 2019. I think I can give you some tips to address this question. So basically, we have to have multiple layers of checklist. So one thing is like there are different evidences and each can help you with come up with different interpretations. So basically, it starts with what type of biological question I'm asking. So when I'm exploring macromolecule assemblies, I would look for anything that has interaction evidences which are collected from assays that has homogeneous experimental setup. And with heterogeneous experimental setup, then I know that they are artificially introduced in the system. They can very well give me more interaction strength, but also can give me value additions like the mutations or the clinical variants that affect the interaction and the outcome, the post-translation modification effect on the interaction outcome, and the agonist and the antagonist list. So basically, it is the good way to start. What type of question are you interested in? Are you interested in anything that breaks in particular interaction? I would suggest you to look for the low throughput evidences. Are you looking for contextual interaction outcomes? So then pick up something that has coming as an interaction outcome of like proximity partners, bio-ID assay or affinity purification mass spec data, tandem affinity purification mass spec data outcome. So based on that, you can generalize your, you can narrow down to a first context, and then like say, if you're picking up in high throughput. So for example, I did in Alzheimer's network analysis, and we had a paper recently published. So the choice that I made was to pick up a big interactome, tau interactome, which was affected by the clinical variant. And so compare it with the wild type interactome, I mean, the canonical tau interactome. And so that led me to come with the differential enrichment of macromolecule assemblies. So this affinity purification mass spec data that gave me two contexts, one that is detected in a particular tissue, which I selected as human neurons. And then it also gave me another context at the perturbation level. So the BAIT protein carrying a mutation or clinical variant of my interest. So you have to navigate between, I mean, the best question is like, what is my question? What am I trying to address? So based on that, you can pick up those, strategies and assays that help you to address those questions. So that's how you go through. That's my going way forward. Yeah.
If I may add just a tiny thing, one strategy, if you want to bring more context and so that your data is more relevant within the specific context of interest, let's say in a given tissue, what you might want to do is to use some baseline expression data to then enrich your network so that you can filter out all the proteins which are not present in the given tissue or cell line of your interest, and can really focus on the interactions really happening between the proteins present in that context. So don't hesitate. That's one of the powerful aspect of Cytoscape. You can easily combine many different data types, including expression data or protein abundance data within Cytoscape, and then filter the nodes based, and so therefore the edges based on their expression level. So that's what I would recommend as well.
Thank you.
Yes, one point. And IntAct also provides you with the confidence level score. Then you know the strength of the interactions that you are assessing. So you know that. Yeah.
Okay, so I think we can conclude the webinar. We're like around 5 minutes past time. Someone is asking one last question. Which are the most commonly used assays in regard to last response provide to check molecular interaction?
Okay, so the high throughput interaction evidences, so we have most of the high throughput assays coming from E2 hybrid, and the next highest evidence is contributed by affinity purification mass spec data. Yeah, these are the most common things that I notice.
If it was related really to my answer on my question, then I would recommend the Human Proteome Atlas experiment as well for the baseline level HPA experiments, which are also hosted in Expression Atlas. That's what we use in Reactome as well.
Perfect. Thank you so much. So thanks again, Susie, Elliot, and Kalpana for joining us today. And thanks to everybody who has joined us for the webinar, has asked questions, and is following us across the month for the different webinars. So we do have one last webinar that is running next week. We'll focus on UniProt. So if you're interested, registration are still open. Otherwise, I'll see everybody next year.